The human stomach is the main site of digestion through the production of gastric acid to break down large food particles for the body to absorb. Parietal cells line the lumen of the stomach, and it mainly consists of enterochromaffin-like cells, chief cells, and G cells. Parietal cells produce and secrete gastric juice into the lumen of the stomach through a sophisticated pathway. Firstly, hydrogen ions are generated within the parietal cell from dissociation of water. The hydroxyl ions formed in this process rapidly combine with carbon dioxide to form bicarbonate ion. A reaction catalyzed by carbonic anhydrase bicarbonate is transported out of the basolateral membrane in exchange for chloride. The outflow of bicarbonate into blood results in a slight elevation of blood pH known as the alkaline tide. This process serves to maintain intracellular pH in the parietal cell. Chloride and potassium ions are transported into the lumen of the canaliculus by conductance channels, and such is necessary for secretion of acid. Hydrogen ion is pumped out of the cell into the lumen, in exchange for potassium through the action of the proton pump. Acid secretion is controlled by three main mediators: acetylcholine, histamine and gastrin, which are produced and modulated by enterochromaffin-like cells. Gastrin, produced by G cells, bind and activate ECL cells to cause the release of histamine. Histamine being the main mediator, binds to histamine receptors located on the membrane of parietal cells and in turn activates adenylate cyclase, leading to elevation of intracellular cyclic AMP concentrations and activation of protein kinase A. pKa activation causes phosphorylation of cytoskeletal proteins involved in transport of the H+, K+, ACPase from cytoplasm to plasma membrane. As a result, this leads to increased gastric acid production. Oversecretion of gastric acid may lead to peptic ulcer. Histamine 2 antagonist or blocker such as famotidine, nizatidine, ranitidine, cimetidine blocks the histamine 2 receptor, which suppress the downstream mechanism, result in decrease in hydrogen ion and chlorine ion secretion, as well as gastric acid secretion. This inhibition of mechanism promote healing of duodenal ulcers, reduce gastric acid secretion, prevent stress ulcer and GERD. Side effects for H2 receptor antagonist is relatively rare. However, some transient effects may include headache, dizziness, skin rash, diarrhea and confusion. Furthermore, long-term use of these antagonists may cause impotence due to erectile dysfunction and gynecomastia, which is also known as man boobs. These side effects are associated with the blocking of H2 receptors located in areas other than stomach. These include the heart, vascular smooth muscle, and the immune system. Blockage of H2 receptors on the heart causes a subsequent decrease in heart rate, as a result, decreasing cardiac output and stroke volume. Apart from this, the blocking of H2 receptors at vascular smooth muscles may also cause decrease in contractile responses of the blood vessels, affecting the overall peripheral resistance of arteries. The exact role of H2 receptors in the immune system is still being studied. However, past research have outlined that H2 receptors antagonists lead to a decrease in cytokines and antibody levels. H2 receptor antagonist, particularly cimetidine, may have potential drug interactions which interferes with hepatic metabolism of certain drugs. These include warfarin, thiopurine, phenytoin and ethanol. Cimetidine acts non-selectively to inhibit the production of an enzyme called 450 cypriot pounds, which assists in the metabolism of these drugs and therefore decreasing the rate of drug breakdown. Due to the decrease in drug metabolism, this increases the possibility of potential drug interactions. Ranitidine is the most commonly used, as it is rarely to interact with cytochrome P450 while having moderate bioavailability and in relative potency compared to cimetidine, famotidine and nizatidine. Moreover, ranitidine has a faster and longer action than famotidine. Nizatidine, eliminated by renal, had effects on relief and healing from duodenal ulcer. Cimetidine has a relatively higher chance of binding to 450 cypriot pounds and as a result can reduce hepatic metabolism of other drugs, so its use have to be careful with patients taking drugs such as warfarin, phenytoin and theophylline that are mainly metabolized through the liver.